Good afternoon, good morning, good evening. It's a pleasure for me to welcome you to this IUIS webinar, where today we're discussing a very topical and important area, which people from across the globe are really excited to know about, and that is genetic risk and susceptibility to SARS-CoV infection, and particularly severe SARS-CoV or COVID-19 disease. So today we're very pleased to welcome as a guest speaker, Qian Zhang from Prof. Jean-Laurent Casanova's group. She was trained as a pediatrician at the medical school in Fudan University in Shanghai. She then went across to the NIH as a postdoc to be trained in Helen Yu's lab and got into the field of inborn errors of immunity. Since 2017, she was initially a visiting scholar, and now she's become a group lead in Jean-Laurent's group and the Laurent Bell's lab, and her focus is on the human genetics of respiratory viral infections. So before we start her presentation, we just mentioned that attendees should preferably be using Chrome for the best experience with this webinar. And if you have sound problems, check that your browser is not on mute. Please ask your questions. There's a Q&A little tab in the box, and you should please put your questions there as a preference, and feel free to do that throughout the beginning of the webinar. Also, we are recording this webinar, and it will be available on the IUIS website on demand, and it should take about 2 days to get onto the website. And then also we'll discuss at the end of this the next coming series. So without further ado, let me hand over to Qian. Thank you very much and enjoy.
Thank you, everyone. Thank you for attending this meeting. And it's really my honor to present our recent discoveries in the type 1 interferon immunity in patients with life-threatening COVID-19. I'm going to focus more on the inborn error part because of the audience of this seminar, but I will also mention a little bit about the phenocopy of this inborn errors of type 1 interferon immunity that we found in the COVID-19 patients. Oops. So the Casanova lab has been studying patients with severe infectious diseases for 25 years. This is not only in COVID, but every infectious disease that we studied, we always notice that there's a phenomenon that is the infections are usually asymptomatic or mild in the general population, but only very few individuals, the minority who are exposed to the pathogen, will actually develop severe diseases. This happens to, for example, bacteria, viral or fungal infections. And our lab has been tested this hypothesis that is the human genetic inter-individual variability cause these difference in their phenotypes. So when the COVID-19 happens, this is even more obvious to everyone. That is, only the minority of people who are exposed to COVID-19 will actually develop severe diseases. So that's why we immediately organized this consortium with Helen Su in NIH called COVID Human Genetic Effort. This is kind of a outdated figure, but we actually have now sequencing centers over in 40 more sequencing centers all over the world, and we're collaborating with over 200 physicians, local physicians who recruit patients and treat patients from almost every country in the world. And when this happened, we take a step back and just wanted to test a very simple hypothesis. That is the inborn errors that we already know to cause severe influenza can also cause severe COVID-19. So by the end of 2019, the lab has already discovered several 3 monogenic causes of severe influenza infections, including TLR3, IRF7 and IRF9, the red boxes here. We also, with our collaborators, studied several other diseases in the same type 1 interferon pathway. That the blue boxes here, they are not known to cause severe influenza, but also can cause other viral infections. And you might notice that all of these genes, they actually play important role in the type 1 interferon responses. So because there are so many similarities between influenza and the COVID, one important thing being not only, because they are RNA virus, they infect the respiratory tract, but also because influenza, because the antigen drift and antigen shift, the serology of influenza changes all the time, and COVID-19 is a new pathogen to everyone. So these similarities lead us to think maybe they share the same immunological pathways in controlling the infection. So what we did was we tested whether patients with severe COVID-19 also carry mutations in these 13 genes. So we recruited, I think, about 600 patients at that time, and we identified 118 rare variants in these 600 patients who had severe COVID-19 pneumonia. These are rare variants, means their minor allele frequency is less than 1 in 1,000 in general population. And we picked only non-synonymical variants. So these are missense or loss of expression variants. We tested every of these 118 variants by functional assays. I'm not going to talk about all the biochemistries today. I'm just going to take one example. For example, the TLR3 variants we found. You'll see the TLR3 little graph in the middle upper part of the slides. We found several variants in the COVID patients. These are the red ones. And then we use a TLR3 deficient cell line and overexpress these variants, and then test the cells respond to poly-IC. We use Y type as positive control, and the blue ones are the non-loss of function TLR3 variants that we discovered before, and use them as negative control. So by this, you'll see the several red variants has the little star on the top, meaning they are loss of function or severely hypomorphic compared to the Y type. So we did this for every gene in the 13 gene list of all the variants we found. This work is not only done in the Casanova lab, but also our collaborators showing here, Vanessa, Tom, and Isabel, because they are experts in several of these genes. So after we test all of them, we discovered 24 loss of function or severely hypomorphic variants in 23 patients. There is one more variant in the number of patients, because one patient actually carried a compound heterozygous variant. So here is the list of all the 23 patients. You can see they're from different countries. They're different age. I think we have age from 17 to 77, and they're both men and women. And what's most surprising to us is that we identified 4 patients with autosomal-recessive IRF7 or IFNAR1 mutation. These are non-diseases, and these patients looks like they have totally loss of function in these 2 genes. So we focus our studies on these patients. We first studied the IRF7. IRF7 was the first monogenic cause that being identified to cause severe influenza infection. The function of IRF7, it's a transcription factor, and the function of it is kind of to amplify the type 1 interferon signaling, and also in the plasmacytoid dendritic cells, you can think it as the professional interferon producer in the blood, and IRF7 is constitutively expressed in PDC, making it the most potent interferon producers. So we first tested the IRF7 in these patients. You can see P1 and P3 are the two autosomal recessive patients, and they are totally lost of protein in P1 and very little protein left in P3. Then we're lucky enough to actually purify PDCs from the patient P1, and we then infect the PDC with either influenza A viruses or SARS-CoV-2 viruses. The x-axis you can see is the control, and the y-axis is the patient. And these each dots on the two gray boxes, each dot represent a gene that's up or down regulated. And you'll see immediately there's in the control there is this cluster of type 1 and 3 interferons upregulated more than 10 times in the control PDC infected with two viruses. But in the patient, these genes are either unchanged or in some cases even downregulated. So by doing this, we show that the patient PDC with autosomal recessive IRF7 deficiency cannot respond to these viral infections by producing type 1 and 3 interferons. This is very surprising because this patient P1 is 49 years old, female living in Belgium. And we took her serum and tested whether she has been exposed to any other viruses. And this is a graph showing here. So apparently she, compared to her family members and other people living in the same region, she was exposed to all the common viruses you would expect for a 49-year-old woman. But she has never been hospitalized for any other viral infections, meaning that she was able to control all the other viral infections until she was exposed to COVID-19. We take a deeper look at her serology to different strains of influenza viruses. Again, she was exposed to all these influenza viruses that her neighbors and her family members has been exposed to. She has never been hospitalized for an influenza infection. That means the influenza infection in IRF7 deficient patients has incomplete penetrance. This is very surprising to us. So after the IRF7 deficient patients, we then take a look at the IFNAR1, autosomal recessive IFNAR1 deficient patients. IFNAR1 is one of the two proteins that makes the type 1 interferon receptor. And although there are many subtypes of type 1 interferons, all of them share the same type 1 interferon receptor, IFNAR1 and IFNAR2. So missing IFNAR1 meaning the cells shouldn't respond to any of the type 1 interferons. And so first is that we took the patient cells, PHA stimulated T cells, and show that P5 is the patient who has essentially no IFNAR1 expressed on the cell surface. Then we stimulate either the T cells or the fibroblast with different interferons, including interferon alpha beta, the two type 1 interferon, and interferon gamma or type 3 interferon, and show that the patient cells does not respond to type 1 interferon at all, but was able to respond to type 2 and 3 interferons. So this showed that the patient cells with autosomal recessive IFNAR1 deficiency cannot respond to type 1 interferon at all. Now here we have shown that the patient cells, the IFNAR1 and IRF7 patient cells either cannot respond to the viral infection or cannot by producing type 1 interferon or cannot respond to type 1 interferon. But is this the real cause of their susceptibility to COVID-19? This is another experiment done elegantly by Jeremy Le Pen from the Charlie Rice lab. So what he did was he took the patient fibroblast cells and transduced them with ACE2 and the TMPRSS2, the two famous the SARS-CoV-2 receptors. Now these human fibroblasts became susceptible to SARS-CoV-2 infection. Then he first tested patients with either autosomal recessive or dominant TOS3 mutation. That is, TOS3 is the RNA sensor in the patients, and he showed that and then he infected the cells with SARS-CoV-2 and stained spike protein intracellularly. So all the dots here, if they are in the up right corner, meaning there are more spike protein and more viral proliferation in the cells. And when he tested TLR3 deficient cells, he showed that the patient cells are not able to control the viral infection, which can be rescued by pre-treat the patient cells with type 1 interferon. He then tested the patient cells with autosomal-recessive IRF7 mutations, and he showed that the patient again cannot control the viral infection. It can be again rescued by type 1 interferon. Finally, he tested the patient cells with autosomal-recessive IFN1 mutations, and he showed that again the patient cells cannot control the viral infection. This cannot be rescued by type 1 interferon because the receptor is gone, but can be rescued by transduced with Y-type IFN1. So by doing this, he showed that the patient cells either cannot detect the viral infection, cannot make type 1 interferon, or cannot respond to type 1 interferon. These patient cells cannot control SARS-CoV-2 infection, and this is probably the immunological mechanism of the disease. So I've shown you that the patients the patients have mutations in 8 of the 13 genes we tested in the type 1 interferon pathway are susceptible to COVID-19. Now, we study these inborn errors not because they are the cause of this disease, because all of these patients carry very, very rare mutations. So these discoveries definitely do not explain why there are millions of people dying from COVID-19. However, we study these inborn errors because they point us the way where to look at. They show us that the type 1 interferon pathway is essential. Because of that, we very quickly discovered the phenocopy of the type 1 interferon deficiency, that is the patients with autoantibodies against the type 1 interferon, and that is done by discovered by Paul Bester, shown the picture here. Autoantibody as a phenocopy of inborn errors of cytokine immunity, this is not a new idea at all. I can show you 3 examples that has been studied for many years. That is, autoantibodies against the interferon gamma, the type 2 interferon, can lead to mycobacterial diseases, whereas the inborn errors of the same cytokine or the cytokine receptor leads to the same phenotype. Inborn errors of IL-17 cytokine or the receptor leads to fungal infection, and autoantibodies to these cytokines have the same phenotype. The most recently discovered was the inborn error of IL-6 receptor mutations that leads to bacterial infections, and autoantibodies against IL-6 leads to the same phenotype. So mutations in the pathway the autoantibodies against the cytokine is the phenocopy of the mutations in the cytokine of these all the receptors. And even the idea of autoantibodies, anti-type 1 interferon is not new at all. It's been known for over 30 years. This was first discovered in patients who are treated with type 1 interferon chronically, and in a small minority of patients with lupus. And the most interesting thing is the patient who had ER mutation. ER is the gene that deletes autoreactive T cells in the thymus. Almost all of them, at a very young age, all develop autoantibody against type 1 interferon at very high level. Although these patients have not, they have their own diseases, but these autoantibody was not known to associate with any viral infections until COVID. When we sequenced the patients from our severe COVID cohort, we found 3 patients with air mutations. And we immediately connect the dots because we already know they have autoantibody against type I interferon, and we already discovered patients with mutations in the pathway. So we immediately tested the patients in our cohort whether all of them have autoantibodies against type I interferon, regardless if they have any mutations. And what we found was that, in fact, over 10% of patients have autoantibodies against either interferon alpha or omega or both. We found no one in the COVID PCR positive but asymptomatic cohort have autoantibodies. The odds ratio is infinity. If you really have to calculate a p-value, it's highly significant. And we show that the autoantibody found in these patients' serum are actually neutralizing. Here's one example by showing that the phosphorylation at step 1 was blocked after the treatment with autoantibody patient serum, and the ISG induction was diminished. And we show that these patients have neutralizing autoantibodies against type I interferon in patients with severe COVID-19. But again, is this the cause of this disease? This mechanism was again shown by postdocs in Charlie Rice's lab. What they did was they take a cell line that's permissive to SARS-CoV-2 infection. Treat these cell lines with interferon alpha will lead to protective immunity against the viral infection. If you also treat the patients with treat the cell line with the patient's serum, you found that the protective effect of the type I interferon was blocked by the patient's serum. And what's surprising to us is not only the patient has very high titers of autoantibody, you'll see here this one red line in the middle. It's actually a commercially available monoclonal antibody against type I interferon, and you'll see some patients' titer are even higher than the monoclonal antibody, showing that these autoantibodies in the blood is really the cause of their disease. What's interesting is that we found autoantibodies in patients who are elder and mostly men. This number here is not very representative of the true disease because at the beginning of our cohort, we tried to select patients who are younger without any comorbidities because we think that it's more likely that we found patients with inborn errors in younger patients with no other conditions. However, we still found that over half of our patients with autoantibodies are over 65 years. This is highly biased because we collected a younger cohort to start with. And also more surprising, actually, if anything, the most important thing in this entire study is that we found 95% of the patients who have these autoantibodies are men. In fact, among the only 6 women we found that had autoantibodies against type I interferon. One of the 6 women is actually a patient with incontinentia pigmenta, the patients with NEMO mutations. And that means she has totally biased X inactivation in her body. She's genetically, you can consider she's more like a man's phenotype than a woman phenotype, at least in the immune system. So this suggests us that there is a high sex and age bias in patients with autoantibodies. So finally, I showed you these 2 diseases, the inborn errors and the phenocopy of the inborn errors of patients with severe COVID-19. How is these 2 mechanisms come together? We finally test the patient's serum during their infection of COVID-19, and we found that although normal patients who had acute infection of COVID-19 has very high interferon alpha in their serum, shown here in the blue dots, patients with a variety of inborn errors or with autoantibodies has essentially undetectable type I interferon in their serum. This shows us again in the patients in vivo, these two diseases share the same mechanism. It also suggests us that we could probably treat patients with type 1 interferon, at least those without mutations in the IFN1 receptors. And we have already treated several patients, in fact, and reported some cases, and they all showed very good response to the type 1 interferon treatment. So finally, I think I hope that I could convince you that type 1 interferon immunity is very important in control COVID-19 infection. We found about 3% of patients in our cohort has inborn errors. That is a small number. However, we found that there are more patients who carry the autoantibodies in our cohort. And all these put together is about 15% of patients. There are still more patients to be studied and more patients to be understood. I would like to thank everyone who worked in a project. We have a huge consortium. Everyone who contributed and the patients and their family should be thanked. Especially, I would like to highlight Paul, who discovered the autoantibodies in a patient. Gigi and Helen from NIH is very important in our patients collection and studying of the autoantibodies, and also Charlie Rice's lab, who contribute greatly to our studies in terms of the viral infection mechanisms. And I invite you to visit our consortium website if you haven't done so, and please follow us on Twitter. Thank you very much.
Thank you very much, Qian. That was a lovely presentation. Please, to encourage the audience here to put your questions up. So I think there are a couple of questions coming through. I'll maybe kick off with one, which is that, initially, as you said, the phenocopy aspect was focused on, you were looking in the younger patients. Have you expanded it now with the growing number of patients to other populations as well? That's, I guess, my first question. And then my second in relationship to that is, 10% is quite actually high. And is there merit, or are there intentions? Have we sort of calculated our number needed to test? So is it sort of worth looking for these autoantibodies? Because perhaps, as you say, maybe we could overcome them with high-dose interferon treatment.
Yeah, so that is a great question. Now, the 10% in that cohort is probably underestimation because our patients are mainly younger. We now extended our recruitment to all ages, all gender, everyone, and this number probably going to increase. And this number is an enrichment of patients with autoantibody with severe COVID. In general population, we tested 40,000 people serum before 2019 to get a real estimation what's the prevalence of autoantibody in general population, and it's similar to what we expected before. It's probably around 0.3%. So that means 3 in 1,000 people, adults, have autoantibodies already. So I think if someone who had, previous viral infection history or has other comorbidities or family history of severe viral infections, or they are elderly men who has much higher prevalence of autoantibodies, should be tested for these autoantibodies.
Yeah, I think for me that makes a sort of next step. Even in, because with the sort of newer variants, certainly our clinical practice here, we definitely started to see, and obviously the higher burden of cardiovascular disease, we started, that's predominantly reflected in the male population. So we know that the mortality rate is worse in men. And so maybe there's a sort of like middle zone of maybe 45 to 65 where testing patients might be advantageous, because certainly I know that the interferon, aerosolized interferon hasn't really, on the general application of it, hasn't really panned out, but maybe there's a place for a targeted use in the context of that, because as you say, that number in the general population, it's actually not too bad. You wouldn't have to test that many people. So I think that's something to consider.
Yeah, and also
Okay.
for the treatment, for the treatment is that most patients with autoantibody, they have it against interferon alpha and omega. Very, very few. I think only 2 individuals we found has autoantibody against beta. And there is a commercially available beta treatment. It's intramuscular. You just one shot. If you know you have autoantibody, you know, I think it is time to really seriously consider to give this treatment.
Yeah, good. So then there's another question coming through here, which relates to STAT1 and T-BET transcription factors, and any data on how they are modulated in severely infected patients, and whether adjusting these factors might help patients.
Yeah, so we are looking for also other genes in the pathway, including STAT1 and T-BET. We so far have not identified a patient with STAT1 mutations, even in a heterozygous model, but we are still looking for sure. The idea is that they probably have more infection at very early age, and they're very careful not to be exposed to COVID. But you know, we're still waiting for these patients.
Indeed.
Yeah.
Good. And are there, do you have any work? Is the COVID genetic consortium at all looking at the multi-system inflammatory syndrome post-COVID-19, the disease focused more on children and adolescents?
Yes, there is a working group in the consortium that focused on this, and Shenying in our group is leading the effort, and Vanessa in UK is also leading this effort. They have actually very exciting new things. Maybe they can talk about it in the next meeting.
In another webinar. Okay, that's great.
Yeah.
And then another question coming through, have you found any anti-type 3 interferon antibodies or autoantibodies? Has anyone looked at that?
Yes, so actually Kai, who's leading this effort, she was the one who actually first discovered the anti-interferon, type 3 interferon autoantibodies in, I think it's the IPS patients. And so she's actually screening for our cohort to look for the type 3 interferon, antibodies against type 3 interferon.
Okay, but no news as well as yet on that.
Yeah, we're still looking at it.
And then I guess maybe I'll just segue to one other question. You know, with the mycobacterial phenocopies, with the MSMD, you know, that was enriched in Asian women. And so it's interesting, like, you know, scanning for these autoantibodies in different populations, I think might also be interesting. Because you mentioned that 40,000. What population group is that? Is that a mixed population ancestry?
Yeah, that's I would say mostly European, because we collected them from France and the US. There will be some other, you know, populations within this 40,000, but not dominantly in Asian population. That's a very good question. Actually, the autoantibody against type 2 interferon is associated strongly with HLA type.
Yeah, great. So yeah, that the HLA might be driving the ancestral association, correct?
It's possible, yeah.
Okay, and then there's another question here in relationship to whether or not autoantibodies could affect vaccine efficacy. I thought that's quite an interesting question, but
I don't think so. There's no reason to think they should. I mean, if the patients also have other autoantibodies like lupus, it might. But in these patients, I don't think so. And we actually suggest these patients to get vaccinated as early as possible to protect them from COVID.
Yeah, and I mean, I think we know from, other patients that make autoantibodies, there's no data. Most of them, unless they're on immunosuppression, that might affect their response to vaccines. Most of them respond to their normal vaccination. So I would agree with you that we're getting vaccinated. In fact, that relates to one question. You know, in the beginning of the talk, you talked about that patient with IRF7 mutation that seemed to have survived to age 49 despite influenza. And I wondered about, vaccination that patient might have had. influenza vaccination. And of course, we've certainly seen with COVID the shortfalls of putting too much weight on neutralizing antibody titers. That cross-reactive T cell epitopes, these kinds of things may have very importance in terms of protecting against severe disease. So I wonder if there's, vaccines certainly stimulate T cells as well, and that's possible. That maybe the patient you're talking about, even though she might have had a deficiency, she might still have had T cell responses to influenza that are protecting her. I don't know. What are your thoughts on that?
Yeah. So yes. So the patients. I don't remember exactly if she every year got the flu shots. That's also why studying flu turns out to be way more difficult than studying COVID, because I think 40% of people are having flu shots protected. Besides, you're also often exposed to lower virulence seasonal flu all the time. So it's very difficult to study. Now, COVID essentially is new pathogen to everyone. Nobody has a memory response to COVID. That actually allows us to study the immunological mechanism easier than other infections like flu. So we're actually learning a lot from studying the COVID patient that can be applied to the flu patients. You're totally right about that.
Mm-hmm. Yeah. And then I guess another question related to sort of treatment approaches to the autoantibodies is has there been any measurement of them after steroid therapy? So, that's now kind of standard practice for severe COVID, high dose steroid therapy, and whether or not that's having any impact on autoantibody. That's an interesting question.
Yeah, that's a very good question. So what we know is these autoantibodies exist before they're exposed to COVID, because we tested the patient serum frozen before 2019. We do think that when they're infected, because the body is making type I interferon, it actually triggers like a memory response. The autoantibody titers can go up during the acute infection, and whether if treat them with steroids during the time can actually inhibit that. That's a very good question. I think we should test it.
Good, thanks. I'm glad you're getting some inputs there too. And then in terms of sort of, I don't know if you did kind of multiplex cytokine measurements in some of these patients, but the question here is that, are there other cytokines that show to be either unexpectedly elevated or downregulated in some of these patients with autoantibodies against type I interferons?
We did in a small portion of the patients. They do have other cytokines made. Their type I interferon is undetectable, but they are making other inflammatory responses that you will expect because they have uncontrolled infection.
Great. Yeah, so this is another question, which is a kind of more general question about maybe gene interactions. It says, why not all of COVID-19 infection inborn errors of type I interferon patients becoming severe and life-threatening? Are there some coincidence or some other factors? In other words, if you carry, I guess the question is, if you carry a mutation, you might still not get severe COVID. Could there be some other, what are the reasons you think?
Yeah, so that's another great question. We are expanding our search in patients, not only in these 13 genes, but in all the other genes. Even within these 13 genes, we are definitely observing incomplete penetrance. That is, you might carry a mutation, but ends up in a more like a moderate or like intermediate phenotypes. And we are really trying to first identify these people. Because before we only look at the totally asymptomatic at the most severe cases, now we're looking at all the phenotype in between, and we are identifying these patients. And I think after we have collected enough of these patients, then we will be ready to study if there's other genetic factors or other epigenetic factors that change their phenotype.
I think we addressed, there's another question about the vaccine. I think we've kind of covered that, whether type I interferons have any impact on vaccination. And then there's a question around treatment. I mean, is there any, this one is around treating COVID patients with type 3 interferons Any data to support trying that approach?
I think that's another good question. What the lab does is we let the human genetic disease show us the data. It's the experiment of nature. I think if we discover that patients with severe COVID-19 and type 3 interferon deficiency, that really shows that type of interferon could control the viral infection, right? So that, I think, will tell us now it's time to consider treat the patients with type 3. So I think it's possible people can try that, but from our angle, we are still looking for the patients to show us the real data.
I agree with that. And then this is quite an interesting question here, which is saying, how is there any correlation of these interferon 1 deficiencies with the hyperinflammation response of severe COVID? So in other words, I guess the question is, in these patients you saw when they got severe disease, did they show the typical hyperinflammation and the typical manifestations of severe COVID?
So that's another great question. I think it's probably the case because when patients have uncontrolled viral infection, you're gonna end up with a cytokine storm. This not only happen in COVID, but in other patients, other infections too. For example, the IFNR2 or STAT2 mutate patients, when they get the MMR infection, they have almost like a HLH spectrum phenotype. So I think that has been reported in these inborn errors, and I will not be surprised if we find these patients in COVID-19 infections too.
Yeah, I mean, I think also the cells, I guess this sort of cells making other cytokines that are driving that hyperinflammation. It's almost like you missed the type 1 interferons, miss the early control, and then the havoc comes thereafter.
Exactly.
So another question here, the patients with anti-interferon 1 antibodies used in the infection experiment also had anti-SARS-CoV-2 antibodies. If they did, how did you eliminate the contribution of antiviral antibodies?
That's a very good technical question. Actually, they do. And what we did was actually the interferon treatment was pre-SARS-CoV-2 infection. And we tested by wash it multiple times. I need to ask Charlie Rice's postdoc how many times he washed, but he showed that this does not inhibit the viral infection by a control serum. So we did test that.
It's a good technical question. I'll keep you on your toes. So this is a comment here. Great presentation. Thank you very much. The challenge with interferon treatment for COVID cases is timing. That's true. By the time the patients are hospitalized, the viral phase tends to be well established. Interferon treatment may not be infected. This seems to be the preliminary data from trials. Nebulized interferon alpha is effective. So I guess that's further comment of what we were talking about too. And then this question is a general question about other cytokine antibodies. You showed those 3 examples, the IL-17, there's several sort of other cytokine autoantibodies. Yeah. Any data from that in COVID?
So in our cohort, again, in a small proportion of the patients, we also tested for other autoantibodies. Some of them might, for example, the APS1 patient, they have autoantibodies against a variety of cytokines, not only type 1 interferon. We have not discovered, for example, like a cohort of patients without any genetic diseases have broad spectrum of autoantibodies. We have not found that yet.
And then was there any correlation with the actual titer value and the severity of disease? So the higher the autoantibody, the more severe, or was there an actual continuous relationship?
So that's another great question. Short answer is that we don't know. The reason why we don't know is here, even the neutralizing assay I showed you is neutralizing 10 nanogram per mil of interferon alpha. That is a huge amount. So in blood, you are in real life, you will not have that high high interferon alpha, you're more like on the picogram level. So what the lab is doing right now is to develop a more sensitive neutralizing assay that we can detect picogram level of autoantibody. So right now, all the positive ones, they are all very, very high to the point that you're just never going to make that much interferon to be neutralized. But we will soon find out if the titer really matters when we can actually detect the lower titers first.
Right. And then this is a question which I think is what's quite interesting. That I think the answer is no, but you can sort of expand on this further. The question is: in patients with autoimmune illnesses, we must suspect that they probably have antibodies to interferon.
Yeah, they must be at higher risk, especially for women with lupus. So we already discovered several patients in our cohort. And in fact, actually, one patient did not have lupus, but then after she had severe COVID-19, she developed lupus after we follow up the patients. So yes, the patients already known to have autoimmune diseases, they are at high risk.
Okay, that's interesting because that clarifies to me, because I kind of felt that the spectrum of patients developing this seemed to be the male, older patients, where I guess, typically in our context, we see younger female patients with more severe autoimmune disease. So I actually thought that answer might have been no. But so there is evidence that the standard kind of younger female patient with autoimmune disease is at higher risk in a similar way to the older male patient?
Yes, yes, that's true. Although still, if you just take all the patients have COVID-19, majority of them with autoantibodies still elderly men. But there is this small peak of young women who at high risk of lupus also have high risk of autoantibodies.
Right.
We don't know why. We don't know why yet.
So I mean, so that assumption is still kind of true, that if you are sort of more prone to make autoantibody, then you are more likely to have type 1 interferon antibody.
Yes, but we still don't know why. Yeah.
Go ahead.
So we still don't know why is there this much higher number of elderly men who has autoantibody and don't have any other problems before COVID-19.
Right. Very interesting to explore. I'm just checking here that there's any other comments here. We talked about that. Oh, there's one question about whether there was any in these patients with inborn error to type 1 interferon. Is there any effect on Th17 cells? And did you look at specific kind of impact on different Th helper subsets?
That's a good question. Again, we have not looked at Th17.
Right, but it doesn't seem, I mean, I guess the early response, as you mentioned, you mentioned dendritic cells, but not specifically on downstream T cell activity, potentially.
Yeah, we have not looked. Yeah, we look very focused on innate and intrinsic immunity so far. In a more differentiated T, Th1, Th2, or Th17 T cell responses, even B cell responses. This actually is another focus of the consortium led by Peter Brodin. He's looking at more patients with so-called long COVID that at the later stage, you would think the adaptive immunity will play a more important role compared to the innate and intrinsic immunity. So that's what he's looking at. Yeah.
Great. Okay, I think we've covered. There's one last question coming in here. Do you have any data about antibodies to interferons in diseases like Sjogren's or rheumatoid? So I guess more into specific types of autoimmune disease.
We have not studied specifically these patients, but if they pop up in our cohort, we will definitely look at them.
Great. And then I guess, well, thanks very much. I think we've had some great questions, so thanks to the audience for doing that. Thanks, Qian, for a great presentation and addressing these questions. How do people, the one last thing I guess is, this is a global consortium that you guys have. How do people get involved? Is there a website, or is there details of that people can make contact?
Yeah, of course. I think in the last slides, I showed the consortium. It's called the COVID Human Genetic Effort. We have a website, and you can just contact the Casanova Lab or me in person anytime if you want to join the lab. There's different format, no matter if you are a clinician or you are a researcher lab or you already have a sequencing center, can kind of have a large capacity. We share data, we share patients, and we share samples within the consortium. And we in fact have once a week, every Monday morning, a meeting within the consortium. So you're very welcome to join us anytime. Just send me an email.
Great. Well, thank you very much. And thanks to everyone to take the time and come and listen. Just a reminder for next week, people can be encouraged to register for the next webinar, which will be on regulatory B cells and the gut microbiota, an intricate acting balance in autoimmunity by Claudia Muari. So we look forward to having people join that next webinar. Thanks very much, and have a good day, afternoon, evening, wherever you are.
Thank you. Thank you, everyone.